So going to the anatomy and histology of liver, we have to first understand the basic features of the liver. Regarding the liver, you know that it is the largest internal organ, and you can also see that the size of the liver is approximately 1.5 kilogram. That is the size of the liver. Now, regarding the development of the liver, embryologically develops from the foregut. That means if you look at the embryo at the embryonic stage, there is during the embryonic development or early development, the intestine is actually divided into three regions: foregut, midgut, and hindgut. Some of the organs will be developing from the foregut region of the embryo, and some will be developing from the midgut, and others will be from the hindgut. So according to the embryological development, we can find that the liver is developing from the anterior portion of the intestine or anterior portion of the gut. That is why it is developing from the foregut. Now, if we look at the structure of the liver or at the anatomy of the liver, we can find four regions in the liver, and there are two larger lobes are present, right lobe and left lobe. And similarly, you can also find two smaller lobes known by the name quadrate and caudate. Here you can see that this is how the structure of the liver. There are two larger lobes are also present here, as well as two lower lobes. The larger lobes are called the right hepatic lobe and the left hepatic lobe, and the smaller lobes are called as quadrate lobe and the caudate lobe. So if you take the liver, the structure of the liver, there are four small lobes are here, or small separate pieces are here. They are left lobe, right lobe, quadrate lobe, and caudate lobe. Here that distinction is not shown here. Here also we can find another structure of the liver. So that is basically the entire liver can be divided into four separate regions or four separate lobes, and these lobes are called right lobe, left lobe, quadrate lobe, and caudate lobe. Then what is the function of the liver? Physiology dealing with the function. Physiology is a science dealing with the function. So going to the functional part of the liver is performing many important functions. Without liver, your digestion may not take place. So basically, liver is a principal digestive organ or digestive gland, because liver is producing the bile. Without the help of a bile, you cannot have any fat digestion in your body. So it is your best friend when you are enjoying some beer with you, because alcohol, the digestion of alcohol, is also helped by or also facilitated by the bile. So this is the why if there is any problem with the liver, the digestion of the alcohol may not be taking place properly. Now here you can find the structure of the liver. Then this is the tissue level arrangement of the liver. Now histologically speaking, each lobe of the liver consists of hepatic lobules. As we have already seen that there are four hepatic lobules, and they are separated from one another by a thin layer of connective tissue called septa or Glisson's capsule. Here you can see that. This is one lobe of liver and this is another lobe of liver. And each lobe of the liver is actually separated from each other by a capsule-like covering called Glisson's capsule. So that is the importance of the Glisson's capsule, or otherwise known by the name septa, to separate the four lobes of the liver. And we have a thin layer of connective tissue. So this is what that blue colored portion is actually a layer of connective tissue which is separating liver into four separate lobules. Then the lobules are visible to the naked eye and are normally hexagonal in outline. Here you can see that this is a one hepatic lobule. So here you can see that if you take the liver, it is formed of different hepatic lobule, and the nature or the arrangement of a lobe or structure of a lobule is usually hexagonal in shape. Now we have to see what are the different portions of this hepatic globule. Now we will go to this is another view of the hepatic globule. Now we will see what are the important regions of the hepatic globule. So basically each liver is formed of number of hepatic globule. Now the nature of the hepatic globule is a hexagonal shaped structure. Now at the center of each lobule you are finding there is a hepatic vein. Here this is a hepatic vein. So if you take the liver, it is formed of hepatic globules. At the center you are finding a vein known by the name central vein. It is also called by the name hepatic vein. Then the cells forming the lobules are cuboidal or polyhedral in shape, and these cells are known by the name hepatic cells. Here you can find there are a number of cells here. These cells are known by the name hepatic cells, otherwise known by the name hepatocyte. Cyte means cell. So hepatic cells are normally cuboidal or polyhedral in shape. Here you can find the different arrangement. These are all hepatic cells. They are cube-like or cuboidal cells are present here. Then and these cells are known by the hepatic cells. Another term is hepatocyte, and they are arranged in radial rows. Here you can find that this is how this is the hepatocyte or hepatic cells, and they are arranged in radial rows. In this picture you can find that this is one row. This is the hepatic vein or central vein, and we can find that different layers of cells are arranged in different radius. So these are known as radial rows of hepatic cells. So that is the arrangement of the hepatic cell or hepatocyte. They are arranged in radial rows of 1 or 2 cell thick rods and trabeculae, which extend from central vein to periphery. Here there is another structure called trabeculae. Here you can find that there is a trabeculae. From the central vein we can find, and there are these are known as trabeculae. This trabeculae is giving the hepatic cells the nutrients and other kinds of materials for its living. So this is how the next region. So going to the hepatic cell, they are normally 1 or 2 layers in thickness, and they are inside the cell. We can find also trabeculae, which is actually connected with the central vein. This is the central vein. Here you can find that. From the central vein we can find different trabeculae coming to the hepatic globules. Then next idea regarding the hepatic cells is that the hepatic cell possesses fine granular cytoplasm, large round central. That is, if you take one cell like this, it consists of fine granular cytoplasm. That is, the cytoplasm will be containing small granules. That is the meaning of the word fine granular cytoplasm, and a large round central nuclei. That is, at the center there is a large central nucleus. Then a deposit of glycogen and lipid. That is, inside this hepatocyte, glycogen and other fatty materials will be present. So that is the meaning of this sentence. Each hepatic cell is having a fine granular cytoplasm, a central nucleus will be there, and glycogen or lipids are also present. And between the hepatic lobe is a labyrinth of narrow irregular space called lacunae. Here you can find that between the hepatic cords. Here you can find that this. Here you can find the lacunae. Which one? So between these hepatic cells, we can find lacunae. Somewhere here I will show you the picture of lacunae. So so between the hepatic cords there is a labyrinth. Labyrinth means winding. That is the meaning of the word labyrinth. Labyrinth of narrow irregular space called lacunae, through which run blood capillaries called hepatic sinusoids. You can also find hepatic sinusoids somewhere. These are all hepatic sinusoids. These red colored portion are known by the name hepatic sinusoids. Here also you can find these are all sinusoids. See, These are the hepatic sinusoid. These violet colored portions shown here are known by the name hepatic sinusoid. They are supplying blood to different parts of the hepatic lobule. So walls of the sinusoids are discontinued and are made of endothelial cells. Okay. Some of the cells become phagocytic and are called cells of Kupffer. Here you can see that this some of these cells. So in the red colored portion known by the name sinusoid, we can find some phagocytic cells. These phagocytic cells present in the sinusoids are known by the name Kupffer cells. Kupffer cells, and their function is they are mainly ingesting, or they are killing the dead cells. They are taking the dead cells like a dead RBC and also bacteria. So their purpose of the Kupffer cells is to destroy the bacteria and also destroy the RBC or erythrocyte. So that is the function of the Kupffer cell. At the corner between adjacent lobules, we are finding strands of interlobular connective tissue called portal area or portal canals. Here you can find that. Here there are three canals are placed together, and that is known as a portal triad. See, this is the three canals are there. One canal is actually bile duct. Another canal is the portal vein. Another canal is a portal artery. So these three tubes are placed together, and they are known by the name portal triad. Here you can say one is a bile duct, other is a vein, and third one is an artery. So one artery, one vein, and one bile duct are placed together, and they are together known by the name portal triad. So this portal triad are usually located at each corner. So this is a hexagonal shaped structure. At each corner of the hexagon, we can find a portal triad. Then each portal, then sinusoids contain blood from interlobular branches of hepatic portal vein and hepatic artery and empty into interlobular central vein. What is the meaning is that the sinusoids will be containing blood from the branches of the hepatic portal vein and hepatic artery, and they are emptying into interlobular canal. I will explain that. Here you can see these are the sinusoids. The blood is reaching this empty spaces called a sinusoid, either from hepatic artery or from hepatic vein, and that will be taken to the central vein. Here you can find at the central there is a vein called central vein, and so the blood is reaching the sinusoid either from hepatic artery or from central vein. Then so these are the basic idea. The liver cell, basic function of the liver cell is to secrete bile. That is the basic function of a liver. So producing the bile is an important digestive juice. It is because of the help of bile we are able to digest the food. And bile is taken into the digestive tract by means of bile capillary. These bile capillaries form bile ductule, which in turn open into the hepatic duct, which ultimately form the bile duct. So this what they are saying is that actually the bile is formed of number of smaller bile ducts. This is the bile duct. These bile ducts are joining together form larger bile duct, and this larger bile duct will be finally opening into the digestive tract. Then in addition to, okay, now we have to see what is the basic histology of the liver. That is, if you look at the histology of the liver, we can find the liver is actually formed of number of important structures. One is parenchyma, another is stroma, and third one is a sinusoid, and there is a space, there is a structure called spaces of Dicey. So we have to see what are the important structures in the liver. One is the parenchyma. Here you can find this is the parenchyma. These cells are known as the parenchyma. Here also you can find that. That is, in the liver, they are formed of different type of hepatocytes. They are known as the parenchyma. Parenchyma is represented by the hepatocyte. So this is a structure of the liver, and each liver is formed of number of hepatocytes. Now the second structure is stroma, which is continuation of the surrounding capsule of glycan. What is a glycan capsule? Here you can see that glycan capsule is actually a connective tissue which is separating different lobules. The connective tissue which is separating different lobules is known by the name glycan capsule. So that is the second type of tissue known by the name stroma, which is actually a continuation of the capsule of the glycan. Now it consists of ഞാൻ പറയുന്നത് കേൾക്കുന്നുണ്ടോ? കുഴപ്പമില്ല, സൗണ്ട് ഒന്നും കൂടെ ഉണ്ടല്ലോ. ഓക്കെ, the second structure is the stroma. Now in the case of stroma, it is എന്താണ് പറ്റുന്നില്ലേ? ഇപ്പൊ കേൾക്കാമോ? And the third structure is known as the stroma. So the first is the parenchyma, and second structure is known by the name stroma. Stroma is actually a continuation of the glycan capsule. Now another structure that we are finding in the liver is the sinusoids. Sinusoids, you can find that these are the sinusoids. Yeah, this is the blood-filled cavity is known by the name sinusoid. Here also I will show you that these spaces are known by the sinusoid. That is the third type of structure, third type of cells that we are finding in the liver. Then another structure that we are finding in the liver is known by the name space of disease. Here you can find that this is another type of tissue that we are finding in the liver, known by the space of disease. So if you look at the structure of the liver, we are finding four different types of cells. One is space of disease. Then we can find sinusoids are there. Then we can also find parenchyma and also stroma. So this is the histological arrangement of the liver. So regarding the histological arrangement of the liver, we can find that you have to explain what is a parenchyma. You need to explain what is a stroma, and you have to also explain what do you mean by sinusoid or space of disease. So this is an idea regarding the structure as well as the physiology of the liver. Once again I will repeat. So going to the structure, anatomy of the liver, this is the liver is actually composed of 4 lobes are there. One right lobe and one left lobe. Then 2 smaller lobes called quadrate and caudate. Then what we have to see is that what you have to understand what is the meaning of the glycan capsule. Glycan capsule acts actually a kind of connective tissue separating different lobules. Then we have to see what is the central vein. This is known as the central vein. Then sinusoids are there, which are simply blood-filled spaces in the lobule. These blood-filled spaces in the lobule are known by the name sinusoid. Normally at the corner of each hexagonal lobule, we can find 3 tubes are there. One is a bile duct, another is a portal vein, and third one is the hepatic artery. They are placed together, so that is why they are known as the portal triad. Then next point is a Kupffer cell. Kupffer cells are actually cells present within the sinusoid. Their function is to destroy RBC as well as bacteria. Then we have to also see what are the three layers which are present in the liver. So this is regarding an idea regarding the structure of the liver.